Welcome, everyone. I'm glad to have you here. I'm speaking to you from southeastern Wisconsin. And today we're going to be talking about maize, corn, and the breeding program that I've been involved in. I think we have 51 growing seasons in on it, breeding for organic farming. And what we've discovered, in particular, the endophyte connection with the corn, that I have been working on with James White, who's also here today. And we're going to talk about what we've gradually learned over time through that breeding program. And hopefully during the discussion, we can get to talking about what the implications are for the crops that you grow. So I think I'll leave it at that. Maybe I should just say I have a master's degree and PhD from Washington State University. I worked 25 years at Michael Field's Agricultural Institute, and then started the Mandaamin Institute in 2011 to continue the work in breeding the corn. And I'll leave it at that. James?
Thank you, Walter. Yeah, James.
Okay, thank you. I'm happy to be here presenting with Walter. He's going to be doing most of the presenting, but I am professor of plant biology at Rutgers University, and I've been here for approximately 27 years or so. PhD at the University of Texas. I actually worked working on endophytes, worked on fossil fungi after that. Then I got my first job at Auburn, Montgomery, and then 95 came to Rutgers, and I've been working on endophytes, first fungal endophytes and now bacterial endophytes. And in particular, what we're looking at is we're trying to understand the mechanisms of basically how endophytes are working. And so in terms of this nitrogen fixation in plants, we're trying to figure out the actual mechanisms of transfer between where the nitrogen fixation happens and how the nitrogen is transferred over to the plant. And we're looking at leaves, we're looking at roots. Walter will talk about rhizophagy cycle, but he'll also allude to some of these other symbiosis that we're looking at. And we've been collaborating with Walter for a few years now, and trying to help him to develop some better nitrogen fixing, organic nitrogen fixing corn. So, Walter.
Thank you so much, James.
Great.
Walter, I don't know if you want to say anything else before I start your presentation.
No, I think the presentation is pretty self-explanatory. I'm sorry it's canned, but it's probably a good idea just to keep on time. So let's go for it, Jared.
All right. So Walter has pre-recorded his presentation. Which in part means that if there are any questions, we'll have time for questions and answers and discussion after the presentation. If you would like to kind of, as you're watching the presentation, just have questions that pop into your mind, you can put those in the chat and we can then have those, a record of those to go through after the presentation. So with that, I'm going to share my screen and hopefully successfully get this presentation rolling.
Hello, my name is Walter Goldstein. I'm Executive Director of the Mandaamin Institute located in southeastern Wisconsin. Today, I'd like to talk with you about corn and seed-borne endophytes. As you probably know, corn is our major cereal crop in the United States, and it's a great blessing. But it also is a major polluter, because corn causes and initiates nitrate production underneath the roots, a process called nitrification, and also because large amounts of nitrogen fertilizers which produce nitrates are needed in order to produce corn. Nitrate from soil and from fertilizers pollutes our groundwater, and also our rivers and our lakes. And it also creates nitrous nitrous oxide, a very potent greenhouse gas, which helps to destabilize our climate. Modern corn has actually been bred to produce high yields, but with nitrogen fertilizers. It's dependent on them. And in fact, recent research done at the University of Illinois shows that modern corn varieties have enhanced ability to cause nitrification and also to cause denitrification of producing nitrous oxide. The problem of corn, as well as the blessing of corn, is not just one for conventional agriculture. Organic farmers also use conventionally bred hybrids and quickly available forms of nitrogen, such as chicken manure, to meet the needs of their corn. And that problem with nitrate is causing problems and harming the country and also the climate. But unfortunately, at present, there really isn't a clear solution. It seems to be an intractable problem in the context of our present social political system and our paradigms. I'd like to share with you today the results of a long-term breeding program carried out first at Michael Field's Agricultural Institute and then since 2011 at the Mandaamin Institute. One of the objectives of that breeding program was to reduce nitrification and dependence of corn on fertilizers. And over time, I've become convinced that many of the breeding results that we've had with that program can be explained by the presence of endophytic microorganisms that are seed-borne. I'll share with you some basic research that has been done by James White and his colleagues at Rutgers University. And also, I'll share with you some of the research that we're doing with organic and conventional farmers in the Midwest to answer some of the practical questions that have to do with corn that utilizes endophytic partnerships in order to be able to obtain its nutrients. And finally, I'd like to address some needed changes in the paradigms that underlie our approaches to agriculture, both as breeders and as farmers, and also as policymakers, if we are to make effective solutions to agriculture's problems. First, let me introduce our breeding program. It's a field-based breeding program. We've been breeding for 51 seasons under biodynamic conditions in Wisconsin. And during the wintertime, we go to Puerto Rico. We had an organic winter nursery for many years, or to Chile with a conventional nursery. Our program is based on making crosses between land races, or teosinte, and adapted modern corn varieties. We use a pedigree-based breeding approach. We produce inbreds. We also produce populations. We do early season testing, so early generation testing, where we test the products of our inbreeding for their ability to produce competitive yields in relation to commercial hybrids. And we do select for protein quality, carotenoid content, vigor, ability to compete with weeds, grain yields, nitrogen, and nitrogen fixation. In terms of timeline and milestones, we began the program in 1989 in response to requests from organic farmers. And for 14 years, we did produce open pollinated varieties. But in 2002, we began producing inbreds and hybrids in conjunction with USDA ARS out of Ames, Iowa. From 2004 to 2007, we began to notice a curious phenomenon, which we called emergent evolution, where lots and lots of nitrogen efficiency and soft kerneled seeds with elevated protein quality were beginning to pop out of our breeding families in unexpected amounts. And we utilize those in the breeding process. In 2009, we discovered nitrogen fixing land races, and we began breeding that trait into our varieties. In 2018, we began trials with the University of Illinois in 3 different states. In 2020, we sold our first hybrids together with Foundation Organic Seed, also known as Foundation Direct. And in 2021, we will be selling our first hybrids that utilize a nitrogen fixing parent. So that's a little bit of the timeline for our work. In terms of the results, our corn is unique. We've generated inbreds and hybrids in populations with greater adaptation to nutrient and weed limited conditions, competitive yields, increased nitrogen efficiency, and grain nutritional value. Also, most of them have soft grain texture. There's often more chlorophyll in the foliage, particularly under nitrogen deficient conditions, densely branched root growth in the topsoil, larger tassels, and greater phenotypic and genotypic plasticity than for conventional inbreds. Here you can see the rooting systems of conventional inbreds from the companies. Up at the top, grown under a nitrogen limited conditions. They tend to have a very vertical, sparsely branched rooting system. And down below, you can see 4 of our inbreds. They tend to be more horizontally oriented and more branched in the topsoil. Also, we seem to have less problems with root rot. We started noticing what I had mentioned as emergent evolution between 2005 and 2006. And here you can see in the cobs these very soft pastel colors appearing of the opaquer kernels, which were enriched in methionine and lysine. Their increase in frequency were very high and not so easy to explain. And here we see yolks of eggs that have been fried at our table at home from our chickens who are feeding on our corn. They're very orange, and that's desired by people for good reason. The high carotenoid content is very good for human health, notably for eye health, but also for other qualities. The carotenoids are very important to help prevent macular degeneration. And those eggs bring a higher price in the store, quite a bit higher. They're viewed as being health food eggs, and they come associated with the higher price. In terms of the grain yields, generally similar grain yields, but somewhat lower yields under high input systems and somewhat higher yields where the fertilizer use is reduced or not used at all. Also, we have found with some of our putative nitrogen fixing hybrids that they respond poorly or negatively actually to manuring with fresh direct applications of manure, which doesn't seem to be the case for our other hybrids. We've carried out trials with the University of Illinois, in Illinois, Indiana, and Wisconsin on organic farms under mostly manure fertilized conditions with funding from USDA OREI. This was done in 2 regions, the Central Corn Belt in Illinois and Indiana, and in the Northern Corn Belt in Wisconsin. 13 farms a year, 2 years, unreplicated strip trials, 2 Mandaamin hybrids on each site, and these were 17.C46 with a relative maturity of 105 day and 17.C2B2 with a relative maturity of 110 days. In these trials, we also used high-yielding commercial checks. The trials we harvested the grain. It went to the University of Illinois for distribution for testing. Some of the testing was done at the University of Illinois. Data were analyzed with an analysis of variance. and were statistically significant. We used t-tests to test differences between hybrids. In terms of the yields, the difference in yield between the hybrids wasn't statistically significantly different. However, the 17.C46, which was the earlier hybrid, didn't do as well in the central corn belt, which could be expected. It wasn't really adapted for that. But it did well in Wisconsin and where it was better adapted. On average, this 17.C2B2 averaged 10% less yield than the Czechs over all sites. That wasn't statistically significant, but nevertheless, I think is pretty indicative of the kind of yields that we see on manured sites. But we also had lower test weight and seed density because they're softer textured flour corns. In terms of the quality, the nutritional value in particular was much better for the Mandaamin corns. The two Mandaamin hybrids had 10 and 18% more protein, 32% more oil, 44 and 63% more zeaxanthin. That's the orange pigment that's so important for eye health. They also had more lutein, 48 to 150% more beta-cryptoxanthin, and 38 to and 65% more beta-carotene. Those are the provitamin A, also very important for eye health. 10 and 15% more lysine, 33 and 42% more methionine, and 10 and 16% more cysteine. The methionine and the lysine are essential amino acids, and they're very important. They're number 1 and number 2 in terms of limiting amino acids for producing poultry, and they're strongly a problem for organic poultry producers in this country who are limited in the amount of synthetic methionine that they can feed to chickens by USDA regulation. All the differences that I mentioned here were statistically significant at the 95% security level. The Mandaamin hybrids also had 23 to 25% higher methionine contents in their protein, which is significant because it's not just that we've increased the protein content in the Mandaamin hybrids. Actually, we shifted the type of protein that they're producing. They have more valuable amino acids in them. And the mineral analysis was also interesting. The Mandaamin hybrids had 17 and 24% more iron, 22 and 32% more manganese, 13 and 54% more copper, and 11 and 12% more zinc in the grain than did the Czech commercial hybrids. Greater nutrient density. Now, when we worked with a poultry nutritionist and we looked at the value, okay, we have 10% less yield, but what about the feeding value of this corn for chickens, simply based on the methionine content difference? And what we found was that actually the conventional and the Mandaamin hybrids had the same value for organic poultry producers because of the higher methionine content in the grain. In summary, the high methionine and carotenoid corn differ clearly from commercial hybrids by delivering significantly more methionine, more carotenoids, and trace minerals at comparable yield levels. Now, let's switch gears for a moment and go to the issue of nitrogen fixation. As I mentioned, we had discovered nitrogen fixing land races, and we worked for many years to try to get this trait into adapted varieties. And we succeeded with the C46 family and the LAT7 family, which you see here on the right. And we're comparing them here on a nitrogen limited site in Wisconsin in 2019. The FN inbred on the left-hand side shows you through its lighter yellow color, lighter yellow-green color, that it is deficient in nitrogen, whereas the C46 and the LAT7 look as if they have been fertilized with nitrogen, but they haven't. And this is very typical for our results on nitrogen limiting sites. Our varieties that we believe are nitrogen fixing look as if they have been fertilized with nitrogen. The LAT7 was bred utilizing a landrace called Milenio, which has weeping brace roots. And here you can see another Milenio derivative with the typical weeping brace roots. The tips actually look at is if they're oxidized, like cigar butts, at the end of the growing season, where the mucoids that are discharged by those roots harbor nitrogen-fixing bacteria, and then the results get harvested by the plant and contribute to its nitrogen budget. The other type, the C46 type, yes, it also has weeping brace roots, but it appears to be fixing nitrogen long before it produces any brace roots. It looks super dark green earlier on under nitrogen-limited sites, quite a bit better than commercial varieties or inbreds. And so there's more at play here, and that's also shown a little bit from our results. Here you can see some results with a wide range of different hybrids based on C46 over here on 5 sites between 2019 and 2021. And we can see the average values for where we did not manure and where we did manure. And you can see there's a 10 bushel difference in yield between where we didn't manure and where we did manure. So manuring reduced the yield. For the NG10 based hybrids, which are not, we don't consider them to be nitrogen fixing, you can see that manuring gave a higher yield, as it did for the foundation organic seed check. You can see that manuring did increase the yields there. So we can ask ourselves, how and why has this happened? And we did. And what was very helpful for us is after we published some of our results in a referee journal article, we were contacted by James White, who has been doing research on endophytes and seed-borne endophytes. And he wrote me and he said that he thought he knew what might be going on in our varieties. And he has worked with something which he calls rhizophagy. And rhizophagy, the rhizophagic bacteria induce rhizophagy or endophytic bacteria. They're seed-borne endophytes, and they can increase mineral nutrient uptake and obtain nitrogen from the atmosphere. It's all based on a biochemical dialogue between the bacteria and the plants, and it takes place in root hairs and in the epidermis and in the skin of the plant, so to speak, in the aerial parts and in its hairs. In short, maize plants could be farmers, farming bacteria, and also selectionists, breeding the bacteria that fit them and then handing it down to the next generation. So a kind of a seed to soil system, which perpetuates itself in different varieties. Here's how the system seems to work. The special mix of selected endophytic bacteria comes from the mother plant. It lives in the seed. It multiplies in the root hairs, in the root tips, and in shoot trichomes, in the hairs. The plant cells attack the bacteria. They foster them, but they also attack them with reactive oxygen. They degrade bacterial cell walls and membranes and feed on that, releasing proteins and minerals absorbed by the plants. The surviving bacteria stimulate the branching of roots and the production of root hairs. The bacteria are expelled through the root hairs into the rhizosphere, where they grow cell walls again. And there they presumably feed on the soil and may be taken up by successive growing root systems of young rooting systems, other ones, that can take them up and utilize them again. It's kind of like milking the cows and sending them out to pasture. Rhizophagy is what this process is called, and it increases mineral uptake by plants. And it's also associated with nitrogen uptake from the air in maize. Here's a picture from Weaver in 1927, old type, very horizontal rooting system. But you can see all the numerous root tip meristems, all these tiny little root tips. And these are the sites where microbes can be internalized and nutrients can be extracted from microbes in the rhizophagy cycle. So how does it work? Well, actually progress has been made on that by White's team. The intracellular bacteria living in cells secrete ethylene. Ethylene triggers the plants to grow those root hairs and to supply the bacteria with carbohydrates. But the plants also produce superoxide, which oxidizes the bacteria and threatens them. In defense, the plants use those carbohydrates they've got from the plant to fix nitrogen, and they secrete antioxidant nitrogen, nitric oxide, or ammonia. And that neutralizes the superoxide and protects their integrity. That nitrate is subsequently utilized and absorbed directly into the plant tissues. Recent studies by White's team have shown also not only that this system is working in roots, but it's also very strongly working in aerial parts, mostly in non-photosynthetic cells, in leaves and bracts, parenchyma, and particularly in trichomes, the hairs. Bacteria could be seen, surprisingly, living in the nuclei of the non-photosynthetic cells, being fostered there. And staining showed evidence for the following in those organs mentioned above: ethylene, reducing sugars, superoxide, hydrogen peroxide, nitric oxide, and nitrate. In fact, all the different chains of the dialogue. The endophytic bacteria in aerial parts were also, their activity was also explored by exposing plants to atmospheres that were enriched in a natural isotope of nitrogen called N15. And those studies showed strong assimilation of nitrogen from the air for a whole range of wild plants, multiple species, and some domesticates, including fescue. Most assimilation was in the aerial parts. Seed inoculated, also they did inoculation studies with seed inoculated with bacteria that they labeled, and they could find that the plants were mostly accumulating those bacteria in their aerial parts and particularly around cell nuclei. And when they did studies sterilizing the surface of the seed before planting it, they found that plants very poorly formed trichomes, the hairs, and there was no bacteria in them and no staining for nitric oxide and nitrate. So the seed-borne bacteria were critical for the setting up of this active nitrogen efficiency mechanism. When they isolated bacteria from the tissues, they found that some of these bacteria were indeed nitrogen fixers, or they could live in nitrogen-free growing media. Here's what it looked like when they did these trials with a glass bell over plants and exposing the plants to elevated N15 rich in atmospheres for a week, and then drying the plants and measuring how much N15 was in them. We see several representatives of different kinds of plants and the amount of nitrogen that was absorbed. We see from the air, we see for fescue, we see for a hosta, which is also a monocot, tree of heaven, and hops. Hops is really hopping here. It's really accumulating. Also hemp. We're very good at accumulating nitrogen from the air. They have built up special glandular systems and chemistry in order to enhance the nitrogen fixing process. Looking at the grasses, studies were done with reed grass, Phragmites, crabgrass, tall fescue, and red fescue. And here is a commonality of what was discovered. The bacteria were indeed living in symbiosis with the nuclei of the plant. They're sheltered and they're released into the cytoplasm through vesicles that come right out of the nuclei of the plant. The bacteria, once released from the nuclei, begin to secrete ethylene, and the bacteria are exposed to superoxide and they respond by secreting nitrogen, absolutely as was described before. The bacteria also, as described before, are in the trichomes, simple trichomes. And also they were found in the epidermal cells, which are kind of serrated in the grasses, these little crooks and crevices in between the cells and the epidermis. So let's take a look at what it looks like. Here's a hosta, and here you can see a nuclei in a cell, and these purple stained vesicles are showing that they're for nitrate, that they're producing nitrate, or nitrates being produced around them. That's quite clear for the hosta. Let's take a look at crabgrass. You see the same phenomena. Here's a nuclei, and here are the vesicles being secreted out of the nuclei with bacteria. In some cases, one can actually see streams of bacteria coming out of the vesicles. That's not clear right here. Here we see bacteria in cells for red fescue, and actually in a nucleus here coming out. Here you can see them in the crooks and crevices in red fescue epidermal cells, which are kind of serrated, and in there these bacteria can abide. And here we see for Phragmites, reedgrass, we see a trichome stained to visualize ethylene production around the bacteria. So how was it with our corn? Well, we sent our corn varieties to James White and his group, and I came over to take a look at when they examined the growing seedlings. And indeed, they all were rhizophagists, and they found, we also sent him conventional inbreds that we had grown side by side with our varieties in the same field right next to them, and they did not have rhizophagy that we could see. But also we saw between our inbreds that they were different in the types of colonization that they seemed to be showing, with more or less spore formers in the roots. And take a look at one of our inbreds families, P40. You can see the bottle brush type of root hairs being formed, which are very typical for the gangs, plants that have gangs of these bacteria in them, and our plants did. It was quite impressive. Here you can see they're pressing out in this primordial root hair just being formed. They're popping out the tip, but also they're coming out the sides. You can see the spore-forming spores being formed here at the endospores. And here you can see a side picture of another root hair, and they're actually emerging right out of the sides of the root hair. Mostly they come out of the tips for most varieties, but for this P40, they were also coming out the sides of the root hairs. In the conventional inbreds, they didn't, the root hair development was retarded. So root hair development is stimulated by these endophytic bacteria living in the plant, coming with the seed. And I should say that the seed was lightly surface sterilized, and the seedlings were grown under axenic conditions. So what we're really seeing is seed-borne bacterial effects. Here's conventional inbred LH206, showing little or no bacteria. And C46, our putative nitrogen-fixing inbred with bacterial aggregates, these sort of clumps of amorphous material mixed in with the bacteria, possible zones of low oxygen associated with the mucosal clumps could help nitrogen fixation. We really don't know. We have to do more research on this. Here we see Novartis 942, a conventional inbred with no rhizophagy, but with endophytic fungal hyphae. And this is a concern. University of Nebraska has reported that most commercial hybrid seed being sold that they found, that they tested, was infected with Fusarium, which is also an endophyte. It's not just a question of endophytes, it's a question of the kind of endophytes. Fusarium does have beneficial effects in protecting the plant from diseases, but it also can produce some horrible toxins, which can render the grain unusable or toxic to animals or people that eat it. So it's a dubious, it's a endophyte of dubious value for humanity and for the corn plant. I've often wondered, why is it that our putative nitrogen fixing inbreds are so hairy? Here you can see C46, very hairy in the tops. It also has very densely branched roots that are very clean. Well, now we come to the part about practical questions, which we're getting as we try to roll out these hybrids for people to test. And the questions are, well, how efficient are our hybrids in terms of getting nitrogen? How much can we reduce fertilization? How well do soil tests predict productivity of our varieties and also their nitrogen needs? What are the nitrogen relationships between organic matter and manures and nitrogen efficient corn? So we've been doing, with help of USDA, NIFA, SARE and OREI, we've been doing, and also with help from the Ceres Trust, we've been doing strip tests and randomized experiments on working organic and conventional farms. And we've compared our hybrids with a commercial hybrid. And we've been looking at different farming systems to see how they affect nitrogen uptake. One of the big issues is not so very easy to come to unequivocal results in terms of, are the plants fixing nitrogen or not? And what we're doing is actually working with natural isotope abundance. There are two isotopes of nitrogen, N15 and N14. And we utilize what's called the delta N15 value. This is critical for this kind of research. N14 and N15 exist in a ratio. There's a lot more N14 than N15, but the ratio is adjusted for that. And naturally, soil has relatively more of the N15 in it, whereas air has more of the N14. Why does soil have more N15 in it? Apparently the bacteria are choosy and they discriminate and they keep more N15 in their bodies, and they release the N14 when they consume soil organic matter as nitrate and ammonia, which have much lower N15 value and are much higher in N14, relatively speaking, and much closer to air, although air is lower than nitrate and ammonia. In our previous studies in 2009 and 2010, we demonstrated that the grain resulting from many of our inbreds and populations had higher delta N15 ratios than did conventional corn hybrids. That suggested to us that the corn was getting its nitrogen from microbial biomass, because microbial biomass is highly enriched in N15 relative to bulk soil, and certainly in relationship to nitrate and ammonia, or if either microbial biomass or extractable organic nitrogen, very high in delta N15. But we also found, when we had the land races that were fixing nitrogen, that in their grain, they had much lower delta N15 ratios, suggesting that they had taken up N14 from the air. We assume that our varieties are undergoing rhizophagy, and that the rhizophagy is enabling the plant to get a hold of that microbial biomass with a higher N15 and higher delta N15 ratio. And that should be especially apparent in roots, where the plants are getting that microbial biomass through rhizophagy. But we also assume that when in the foliar portions of the plant, where the plant is getting N14 directly from the air, that we would have a dilution effect of what's coming up from the roots, which is higher in N15. And so this would be especially apparent in the tops and especially in the grain. hence, we have to presuppose that the plant is involved in a kind of a balance between getting more N15, the plants that have these endophytes, getting more N15 from the soil, from the biomass, and getting more N14 from the air. So the delta N15 ratios are kind of a compromise between enhancement of both of these microbial/plant processes. And that overall balance may shift from year to year, depending on whether rhizophagy predominates and uptake of microbial biomass or easily available nitrogen, which comes from decaying microbes, or whether the plant is getting more nitrogen from the air. Now, we made a study, and this is one of many studies, but this one is one we've recently analyzed, and it gives us some very clear results. The study was done for 3 systems on farms that represented those systems, 2 farm sites in each of the 3 systems. The arable organic was centered on production of crops with inputs of poultry manure. The cattle-based organic system was where there was production of beef or dairy, and forages were a major part of production. The conventional monoculture was a dairy system where large quantities of slurry were being applied to monoculture corn. So we took soil samples in the spring, particularly based on organic matter evaluation, and in the fall we took samples again, looking for parameters relating to nitrogen using a different set of soil tests. And we tried to get an idea of how predictive were these soil tests of what actually happened. We tested 4 hybrids. We tested FOS 8500, a conventional very high yielding hybrid. And we also tested 4 of our hybrids, the C2B2, C46, which we predicted might be our most nitrogen fixing hybrid, 17.461, which we also thought was fixing nitrogen, and 17.2B24, which we considered to be nitrogen efficient, but perhaps not fixing nitrogen. And here you can see the C2B2 by C46 hybrid on the Zinnecker farm in Wisconsin a couple of years before we began this experiment. It's very robust, very dark. It looks like it's fixing nitrogen or something, or getting nitrogen in some way, relative often to many conventional hybrids. In terms of the soil characteristics, the samples that we took at planting time showed that the 2 sites in the arable organic systems did have higher levels of available phosphorus and potassium than the cattle-based organic system. The 2 sites in the cattle-based system, however, had higher levels of total carbon and nitrogen, higher carbon to nitrogen ratio, greater amounts of potentially mineralizable nitrogen, and higher amounts of particulate organic matter. So they had soils that may not have been as mineral rich, and of course, poultry manure is very rich in minerals, but they had built up soil organic matter and soil organic nitrogen. And when we looked at the correlations between what happened in the field and also between the different soil tests, we had the following results: soil organic nitrogen and the carbon to nitrogen ratio, and also soil protein seemed to be, and structure, seemed to be key factors that were affecting the yield and the nitrogen uptake of the crops across the board. The spring and the fall tests correlated. And we found that the soil organic carbon and nitrogen and the C/N ratio correlated very well with soil protein. It was remarkable. 99.6% of the variation could be explained. And protein score, and 96% for SLAN, which is an indicator of amino nitrogen, which is another quick nitrogen test, and for nitrate, and even carbon dioxide respiration. And we modeled the performance of the crops and how was it affected by total organic nitrogen and carbon to nitrogen as covariates. And we found that quite substantial variation in the performance of the crops could be explained by that combination of total organic nitrogen and carbon to nitrogen in the soil. 36% of grain yield, 57% of stalk yield, 51% for roots, 45% for nitrogen uptake, 66% for carbon production, 45% for tissue, percentage nitrogen, etc. Also for delta N15 in the grain, quite high. Yes, there was a correlation between the good old-fashioned total nitrogen and carbon organic matter measurements and the newer tests for soil quality, but it seemed that it was mostly soil protein content and soil structure that correlated with the productivity of the crops. One of the very interesting findings when we in the fall, we looked at the soil underneath the roots of our different varieties was that the Mandaamin hybrids had less nitrate under their roots in the soil, about 18% less, indicating possibly less nitrification in the soil under those hybrids. We need to check this out. This is major. When we looked at yields, we had about 11% less yield for the Mandaamin hybrids in the FOSS in terms of grain. We had 12% more stalks, 24% more roots, and lots of nitrogen was in those stalks and roots for the Mandaamin plants. Possibly that would, of course, help with the stalks and possibly help with the silage quality. In terms of mineral uptake, per acre, for the microelements, we had between 14 and 20% more total microelements being taken up relative to the FOSS 8500. So considerably more uptake. It wasn't always significant in terms of individual nutrients, but as a whole, that was the tendency. And for macro elements, it's more or less the same picture, between 5 and 19% more minerals being taken up with the Mandaamin hybrids than with the conventional hybrid. And there seemed to be a correlation between the delta N15 values, which indicate what's going on with the nitrogen and the isotopes and nitrogen fixation, and grain dry matter, grain carbon, stalk dry matter, etc. There seem to be some pretty strong correlations for the systems by hybrid interaction. Also, for mineral uptake, there seemed to be a negative relationship. The lower the delta N15 ratio, indicating more fixation, the higher the mineral uptake per acre. So the two processes of nitrogen fixation and mineral uptake seem to be coupled. Now let's look at the delta N15 ratios. Here you can see for the FOSS 8500, the conventional hybrid. Here we have the root value, the stalk value, and the grain value. And the difference between those is not so very large for the FOSS 8500, but wow, if you look at something like the C2B2, C46, and here are the values of the delta N15 for root and stalk, and wow, is they're much higher than for the FOSS 8500, suggesting a greater uptake of microbial biomass or easily available nitrogen from organic matter. And then in the grain, we see much lower than for the FOSS 8500, indicating nitrogen fixation. And the difference between these two indicate the dilution effect of the N14 coming possibly from the parts of the plant that are fixing nitrogen. The 17.2 B24 and the 17.461 were somewhat intermediate. So we estimated nitrogen accumulation from organic matter and from nitrogen fixation based on those delta N15 ratios. And we assumed that the delta N15 content of the roots represented the isotope composition of nitrogen derived from the soil. And we assume also that the roots of the Mandaamin hybrids are appropriating more delta N15 enriched nitrogen from microbial biomass and from easily available soil organic nitrogen. We also assume that the FOS 8500 took up more delta N15 depleted nitrogen, so less N15 in it, in the form of nitrate and ammonia. And we assume that the difference between the highest delta N15 nitrogen achieved in the root or the stalk and the delta N15 found in the grain indicates a dilution effect due to nitrogen fixation and the amount of nitrogen derived from the air. So here are our estimates, and they're substantial, and there's some fairly clear patterns here. Here are 3 different farming systems: arable organic, cattle-based organic, conventional cattle, monoculture. And interestingly enough, the fixation rates were the highest for the conventional. If we look at the individual systems and how the hybrids performed within them, the C2B2, C46, which we had assumed was our highest nitrogen fixer, seemed to be producing the most nitrogen from the air under the arable organic system and the least under the conventional cattle system, intermediate here. Whereas the other 2 Mandaamin hybrids seem to be producing the most under the conventional cattle system and the least under the arable organic system. When we look at how much nitrogen was obtained from microbial biomass or easily available organic material relative to the FOS 8500, based on the natural isotope abundance, we saw a somewhat similar pattern. But let's look at and here we're making comparisons with the FOS 8500 as our base. And here you can see with the C2B2, C46, yeah, once again, the most under the arable organic system and the least under the conventional cattle monoculture system. Whereas with the cattle-based organic, it differed somewhat in that the most was in the cattle-based organic. That pattern seemed to be fairly similar to what we saw with the micronutrient uptake. Here we see with the uptake of these elements here, on average, once again with the C2B2, the most was in the arable organic system, the least was in the conventional monoculture system, and for the other 2 Mandaamin hybrids, the most was in the cattle-based organic system. And for macronutrients, well, it wasn't quite as clear. In general, the most was in the conventional monoculture system, where lots of slurry, a history of a lot of slurry application, whereas, and then the second most in the cattle organic system. Now, to summarize these, I say, preliminary results of these trials, I have to say that soil organic carbon and nitrogen and soil protein content, the carbon and nitrogen ratio, they seem to be important determinants and tests for predicting crop nitrogen uptake and yield for all the hybrids. The Mandaamin hybrids yielded 10 to 11% less grain, but they produce more stalks and roots than the conventional hybrid. The Mandaamin hybrids took up 5 to 19% more macronutrients and 14 to 20% more micronutrients than did the conventional hybrids. Also, the Mandaamin hybrids obtain their nitrogen differently than do the conventional hybrids. Nitrogen fixation and nitrogen and mineral accumulation from soil were greater for the Mandaamin hybrids and for the conventional hybrids. There was less nitrate also under the Mandaamin hybrids, suggesting less nitrification. and the Mandaamin hybrids differed in their performance in different systems. The plants that were grown in the conventional system had the highest level of fixation, but the lowest level of nitrogen accumulation from soil microbial organic based, easily available organic matter. The C2B2, C46 variety, which we had considered to be our best fixer, appeared to be the best hybrid at fixing nitrogen and at obtaining nitrogen from soil organic matter, but it did relatively poorly under the conventional system. All right, so what does all this mean, and how might it influence how we move into the future? I think that there are some necessary paradigm shifts that might occur, hopefully will occur, or will be considered as we move forward to do more testing and more research. Low nitrogen input, moderate stress for breeders might be a valuable way to go in terms of evaluating crops or for selecting crops because they may enhance opportunities for emergent evolution, which is driven by the corn plant or other cereals or other crops. And the kind of evolution of partnerships might be a hotbed. Corn is also, as we've seen, doing creative parts of the breeding work itself through emergent evolution. It's adapting to environments, it's shaping creative partnerships. And we as humans can select useful partnerships, but their capacities need to be continuously defined and refined by fully engaging and learning from their performance. These may not be static systems. They may continue to evolve, and we may need to work with them. So basically what we're talking about is not really a top-down system, which is usually what breeding is. It's rather a system where we work with the plant and respect their ability to be creative and play the human role in the process in an adequate way. Now, paradigm shifts for farmers. If we bring these endophytic partnership plants out, they're not only getting soluble nitrate and ammonia, they probably are obtaining microbial biomass through rhizophagy and also extractable organic nitrogen from the soil. They may also be obtaining nitrogen from the air, from aerial organs or even from roots. Also, it seems that the productivity of these plants depends on soil building of soil organic matter and total nitrogen. Those may be the critical determinants of the yield for corn if these plants have effective rhizophagic systems. And also, we need to consider how we manure. Nitrogen fixing hybrids with C46 as a parent, they seem to respond negatively to being manured directly, but they seem to respond positively to soil building, to building of soil organic nitrogen. So perhaps we shouldn't be applying the nitrogen to such crops directly, but applying it to other crops, such as grass or other preceding crops or soil building crops. Maybe we need to change our philosophy of manuring. Now, all that being said, there's one other paradigm that's really important, and that is the social framework within which we grow corn. How are we going to introduce such things? We have a problem. This nitrogen problem is not going away, and it's hurting us all. It's hurting the climate, it's hurting the planet, and action needs to be taken. So for policymakers, what kind of policy would make sense to reward farmers to grow such crops, even if they yield a bit less, and to reward farmers that exchange use of partnership climate-friendly corn for which perhaps has a lower yield, for reduced use of fertilizer? How about that? Now, I'd like to acknowledge that this research is built with the help of a lot of people, many organic and biodynamic farmers, the Corn and Soil Health Project at the University of Illinois, USDA ARS, Foundation Organic Seed, Rutgers University, University of Wisconsin Extension, and NRCS, the Mandaamin Institute, my colleagues there, and has received funding from USDA, NIFA, OREI, and SARE, and the Series Trust. Thank you very much.
Wonderful. That was an fascinating presentation, and I would at this point like to open this up for questions and discussion on this topic. You can either raise your hand and I will unmute you, or you can type any questions you have in chat. Guess I'll take advantage of this and ask a question that I had, which was that for seed producers, farmer breeders, other researchers wanting to explore or increase the amount of capability for these kind of beneficial relationships to arise when you're developing or improving or selecting varieties. I know, Walter, you had mentioned sort of selecting in a low nitrogen or moderate stress environment. Do you have any other advice on how you can incorporate this into your breeding?
Yes, and I think James will probably have some ideas also in terms of endophyte populations. From the plant side, I can say that selection probably plays a big role, and it is possible to probably select for the types that look like they may be harboring endophytes. Selecting under conditions that are nutrient deficient. Selecting for very vigorous plants. Of course, sometimes you have outcrosses and so on, and that can complicate things, but for selecting for plants that are unusually effective at thriving under low, or let's say nutrient limited conditions, I think that's a good one. Also, hairy plants. James has woke me up to this, that many of the plants that have trichomes, that have these very hairy leaf parts or leaf sheaths and so on, often many of these plants, at least in the wild species, that they may be harboring endophytic bacteria. Big question for me is seed treatments. If you are getting seed that has a history of fungicide use, there is some literature out there that fungicide use is associated with a diminution of seed-borne endophytes. And as I said, I think that when we're selecting out there, people, even when you're producing seed, if in the seed stocks that you keep back, if you can just keep doing the ones that look the best under your conditions, that look like they're getting nitrogen, particularly if it is a condition where you haven't flooded the soil with a lot of manure. It might be very useful to hang on to those plants or the seed of those plants and allow them to continue to intermate with each other. Those are some thoughts, Jared, but I'd like to, if you don't mind, turn this over to James for discussion of the endophytic communities.
Yeah, I will, and I'll before I go into much about that, I want to mention there is another question by Cooper Brossi from Ernie's Organics, but the question is, are you breeding for better interaction with more productive interaction with soil biology and plant biology? And I think the answer for that is yes. And I think I think that Walter has developed a breeding, and Walter and company at the Mandaamin Institute developed a breeding program that is unique in looking for these natural sources of nitrogen that the plants can then acquire. And what we've been trying to do is to look at plants and specifically look at corn as corn develops and figure out exactly where on that plant that nitrogen is being produced. And the idea being that Walter and Julian, who's here actually visiting us from Mandaamin Institute to try to learn some of the techniques, can then look at these plants as they develop and identify where these microbes are fixing nitrogen, just with a simple microscope or chemical indicator solutions, for example, so that he can select the plants that are doing that process more. So it's a kind of a partnership with a bunch of lab scientists and general kind of plant biologists and a corn breeding people at the Mandaamin Institute, trying to develop something that will some cultivars that are going to be cleaner for the environment than and maybe healthier for people. Some of these nutrients are really useful, and so there's all kinds of beneficial effects that the endophytes provide to the plants, and all those are going to come as Walter and other plant breeders develop more plants that are that have more endophytes and utilize those endophytes more readily. So I don't know if I answered the question, but I talked a lot. So.
Well, but in addition to that, I'd like to say that there's probably people out there who are be interested in this, and might want to interact in terms of doing selection work and checking out whether or not they're working in the right direction. And we're very interested in that, and would be happy to correspond with anyone out there who is interested in selecting in this sort of partnership type of approach. And as James just said, it's a partnership on several different levels. We're partnering with James and his crew of fantastic graduate students who seem to be churning out this incredible information. But also we're working with the plants and with the microbes in a different way. I would say in a more cooperative and a more open way than is traditional. We're not trying to force things here. We want to work with the plants in a way that that indicates a kind of a different paradigm. And I'd like others to try that paradigm.
Yeah, that's interesting. I just saw a question pop up regarding Huitlacoche, the corn smut, right? Huitlacoche, the edible, the eaten corn smut. Yeah, and Julian was just telling me here that some of these hybrids seem to be showing more Huitlacoche, and it could be an association with bacteria. We don't know, but it's really an interesting question, and we don't know. In theory, when you have more bacteria, that would reduce fungal growth. So in theory, we should have less Huitlacoche. So something is not working according to theory, right? Which happens a lot in science. So anyways, yeah, no, that's a really interesting question.
We've had this hypothesis, and it's been based on observation with smut, that we see in some of our varieties that they're more susceptible to what seems to be smut lesions on their leaves, but that these lesions are consumed by the plant. And there are two groups of scientists who've been working with Ustilago matus and claiming that there's evidence that nitrogen is being fixed, possibly in association, as James pointed out, with bacteria, but that this may also be a mechanism or a partnership that enables the plant to obtain more nitrogen, and certainly hormones.
That's really interesting. We haven't gone deeply into that, but there is this close association between bacteria and some fungi. And so if you have a fungus on a plant and you have nitrogen fixers there could be a linkage there as well. So we just have to look closely at that and see if that pans out or exactly what's going on.
We have a question in the chat from Nick Wren, who wants to know, have any of the research plots tried composting manure prior to spreading it out in the field? Some people argue that spreading fresh manure has negative effects on soil microbiology.
This is a really good question, Nick. And this is something I really think we need to do some research on. How do we utilize manure? Is it the same if we compost it? I mean, really compost it well, and then apply it to the field, as if we use fresh manure? Our trials have been done so far with fresh manure, and we do see an apparent negative effect on the C46 containing hybrids. We need to do that kind of research. We haven't got to it yet.
So real quick, I'll add that not all microbes are 100% beneficial to the plant when they're taken into the roots. Some microbes, if they're not adapted to the plant, if they happen to be very resistant oxidatively, they will actually go into the plant root cells and they'll not degrade. They will kind of overwhelm the plant. We call that endobiome interference. Typically in nature, you don't have that because you have lots of other competitive microbes in there, and the plant is getting the best ones. But in the case where you have raw manure, green manure, you may have so many different microbes that the plant is really overwhelmed. And it's a temporary effect, likely, that you put it on, and plants may not grow as rapidly, but as they continue to grow in the green manure, the plants will then start picking up and growing more. And we think what that's about is that some of these inhibitory bacteria get knocked out, right? They're no longer taken in. They go through the system, and then the plants are getting the best ones, and they're growing much better with those others. So there is this phenomenon you noticed, and Walter's told me about that. Other people have told me about this. Green manure is filled with many, many different microbes, and that we think is overwhelming plants with the green manure. Anyways.
Cooper said, in following up on Nick Wren's question, that's the question around the green manure. If I recall correctly, the work of Christine Jones, your results are consistent with some of hers, i.e. that application of N, even manure or compost to crops may not be needed, may not be appreciated by all crops and all soil life.
Yeah, and that's so interesting, Cooper, because the conventional corn and a lot of our corn also does respond to that manure. So I think so the C46 hybrids don't, and so I'm assuming that there is some sort of competition here that's going on, or some sort of James called it, I think, endobiome interference. Was that right, James?
Yeah, that was the term we applied, but yeah, of course we made that up. So yeah, term.
And that may have to do with the established microbiome in these seeds. We find, for example, that if we inoculate something like C46 or C46 hybrids with nitrogen-fixing bacteria, we often have a negative effect. They don't need it. They don't need these bacteria. They have their own community. They have their own colony, their own culture. If we start thinking that the seeds that we breed and select have their own culture, the whole issue of terroir gets a whole different perspective.
And there was another question regarding seeds and seed treatments and the loss of seed communities, and we've done some work on that, and it's absolutely, I'm quite familiar with that area. There are loss of microbes from seeds, the natural community, just from the process of collecting seeds and domestication, and cleaning seeds, for example. Seeds that are gathered as soon as the seed is mature, brought in, put in a cool, dry place in order to store for the next spring to plant them. Those seeds are losing microbes. The natural course of a seed in nature is it stays outside. It's subject to weather, differences in temperature, maybe some soil falls on it, But there's this continuing development of the microbiome. And when we, for many of our crops, we take those seeds early and then we take them in and store them in dry conditions, the microbiome doesn't develop properly. So there is this loss. And there has been studies where people have shown that after a few years of bringing a plant in from nature into cultivated situation, they actually do lose microbes, and then you start to see diseases popping in. There was a paper in the proceedings of the National Academy of Science having to do with some wild tobacco, where some investigators were growing them and brought them into cultivation in Utah, and they found this disease. And they found in the wild plants, they still had those microbes. They took those microbes, put them in their wild tobacco, and cured their disease. So it was pretty good evidence that these plants in cultivation are losing microbes. So basically, we have to figure out how to handle crops in such a way to preserve the microbes, and how you select them in order to breed them, in order to preserve those and maximize those microbes, make them grow better with the soil biology, as one of our, as Cooper, I think, pointed out, or somebody pointed out. John.
Yeah, I'll just add to that in that there have been now some really good studies done with domestication of our cereals. And basically what they seem to be showing is whether it's corn or it's the wheats, or the series of different wheats from the hulled wheats going to domesticated wheat, or if it's barley, that generally we're losing a lot of the endophytic species and races in our seeds. They're declining, and many of them are of the types that would protect the plant from diseases. And so I think James is on target in pointing the direction that perhaps many of the diseases that we are experiencing with our crops have also to do with the endophytic populations that we have lost inadvertently.
So kind of quick follow-up just for everyone who's not looking at the chat. Cooper pointed out that the related to the question of the green manure, that Robert Dungan of USDA ARS in Southern Idaho has since long-term studies on raw manure versus composted and the soil health implications for soil carbon, et cetera. I think related to this question around these endophyte populations, Sean, and I apologize if I'm mispronouncing your name, Agnolo wanted to know, what does this mean for rotations?
Walter, you want to handle that?
Yeah, sure. I think actually James has done some very interesting work with these endophytes that are secreted by different plants and how they interfere with other plants. And I think he was alluding to that also with green manures, or certainly with, I don't know how many of you have had experiences with red clover, for example, as a green manure, about how a number of different plant species, you really have to be careful that you allow this stuff to decompose well enough. And there is information out there, older information even, that indicates that there's bacterial populations from the decaying red clover itself, which then interfere with the growth of corn. And that's the case. I think I've seen stuff from James with carrots and all this companion planting stuff. It may have to do with these endophytes. James, can you follow on that?
Yeah, that's that endobiome interference. And, we can take some endophytes that promote the growth of some plants, take them and put them in other plants, and it'll suppress the growth. Okay, so that clover idea might be that phenomenon. We're just not sure, but quite possibly it is. On the other hand, there is something that is true, and that is you do want to maintain. This is sort of a regenerative agriculture tenet of regenerative agriculture. Okay, you do kind of want to maintain cover crops on your field when you're planting, because that keeps your microbial community in the soil alive. Now, that doesn't mean that you might have something coming out of one plant that reduces the growth of another, but there may, if there's any kind of a time separation between harvesting your cover crop and then planting your corn or whatever you're planting, I mean, that might be enough for that community to go down to a baseline where it's not inhibitory. I don't know. You have to kind of experiment with that. But cover crops are definitely very important for keeping soil biology going in the soil. That's a good point that somebody made. Cooper.
Yeah, for example, also with oats, people grow oats as a cover crop, and it's known as a very benign rotational crop. But in South America, they even grow a different species of oat. They grow the black oats, and the black oats are well known to have effects of cleaning up the soil from pathogens and having just a beneficial effect on other crops, whether it's root exudates or it's the microbial communities that live there. So just being a little bit choosy about what cover crops one uses and what kind of sanitizing or health bringing effects they can have in the soil is also something to be thinking about.
So I have a question kind of related to thinking about this in the context of seed production. So if these communities are kind of developing or have a certain amount of specificity towards the varieties that they're being, kind of co-evolving with as you're selecting them in one environment, do you have a sense of if there's advantages or disadvantages, especially with so often as we move from kind of, unless it's someone selecting for themselves, on their own farm and producing their own seed year after year, there's this movement of seed from, maybe from a breeder to stock seed production to, a larger grow out to the, seed, purchaser and user. As these seeds move between these environments, is there a risk of kind of losing some of those endophyte populations, or is there actually maybe some benefit in picking up additional ones as that seed kind of travels through different environments?
Walter, you want to try to answer that?
Okay, what a question. I mean, it's a good one. And I have been pondering that too. I mean, we send our seed down to Puerto Rico or Chile during the winter time, and it comes back up, but it still seems to be doing its thing. So I guess there's some stability in the communities that we're using, and they've even gone through conventional. I try to get people, when they do it conventionally, not to use, to really reduce their nitrogen use. But we're just in that situation that we can't always find an organic nursery in the wintertime for multiplying our seed. But I think there's a certain robustness there from my experience, Jared. But we really need to, James, pick up on that. What do you think?
Well, I think we have to see, because we have some evidence that very, very dry environments for grass seeds, I'm thinking, because we do a lot of turf at Rutgers, but very, very dry environments, oftentimes seeds are produced out in Idaho or somewhere where it's very dry, right? Perfect for having low disease and so forth. All the water is from irrigation and you don't have much rain and so forth. But the problem is that seed in that dry environment is very dry, and that the dryness will reduce the development of the microbiome on those seeds. It will reduce it so that the seeds then that are produced in the dry environment will not have the same microbial community, or they'll have a much reduced microbial community than those produced in a moist environment. And it so happens, Walter, you're using Puerto Rico, which is a pretty moist environment.
Yeah, so for sure.
You've got lots of stuff so when you make your corn, those microbes are still able to get moisture and develop on those in that plant or on the plant, on the seed. In some cases, they go inside the seed. In other cases, they're replicated on the seed. So, or vectored on the seed. So, so the environment may be important. And, that's what we're trying to figure out. What we're doing is very, kind of interactive to figure out how to Walter is breeding the plant, but we're trying to, figure out what you look for to find the best nitrogen fixation and so forth, and where in the plant. And, the same question goes to now, where do you produce your seed? That might be important, and so, anyways, yeah, it could be very important. And seed treatments, of course, would be very important. Anything that sterilizes seed is not a good idea. Yeah, anyways, yeah. Did I answer it good enough?
Yeah, that's very helpful.
Okay.
Thank you. Nick Wren says, James mentioned the importance of cover crops. Along the same lines, do either of you have thoughts on using no-till methods to help endophytic populations survive in soil systems?
It's better. Yeah, no-till is better. Yeah. Deep turning is the worst thing you can do. Destroy the whole soil community. Deep turn to kill your bugs, right? And then you end up killing all your microbes and putting your topsoil down underneath. And the whole microbial community is buried under a foot of soil or something like that, So yeah, no-till is better.
I'd love to do some research on that. And really, we often do plow. I hate to say that, admit this, but the truth of the matter is that sometimes in order to select under low nitrogen conditions, we follow after grass, which immobilizes a lot of nitrogen, but doggone, you got to kill that stuff. And so a moldboard plow actually does a very effective job of doing so. And yeah, it's probably true, but we really do need to try it and see how things actually work.
Yes, right. Exactly. Most of my work is done in the laboratory in Petri dishes, right? Or in a greenhouse and plants. So I don't have a whole lot of experience in actual agronomic things. So,
right.
I had one course back when I was an undergraduate in agronomy, soil science and so forth. So that was
good for you.
Yeah.
But it does bring up the question about dealing with commercial fields that have been conventional and have Roundup residues. We don't, there is some literature that Roundup kind of dissuades certain categories of microbes. And this is a question I get from the conventional farmers that we work with. Will nitrogen fixation work in a Roundup ready corn bean system? And that's another area where we just don't have any good research. What's the effect of the herbicide?
Yeah, that's right. Some of these herbicides are antimicrobial too. I think glyphosate was initially, I think I read somewhere or someone said that glyphosate was a possible antibacterial substance early on, but they found out that some bacteria could actually degrade it. And fortunately, they found this other effect around the glyphosate actually affecting plants. And so, hence it's not an antibacterial, but an anti-plant.
So it is now been an hour and a half. Does anyone have a final question for our presenters? And if not, Walter and James, do you have any parting words?
Simply, to those of you who are interested in this subject, feel free. From my standpoint, I can say to contact me, and perhaps we can dialogue more.
And thank you. I really enjoyed being part of the presentation, and all the questions are really thoughtful and very, very helpful. So thank you very much.
Yeah.
All right, we do. I'm just going to give one last question because I said it, and Beth Everett wants to know, what about using soil as a seed treatment?
I think that's a great idea. Walter, go ahead. I'm sorry.
No, I found that in certain farms, when you grow the seed there, that the next year, I'm suspicious that we have some sort of benefit coming from those. In particular, there's really one long-term, I think the oldest organic farm, or one of the oldest ones in the country, and near us is Zinneker Farm. And after growing seed there, I just, whenever I do it, I see things seem to almost pick up. And I can't prove that, but I would like to. I would like to see what is the effect of different farm soils on the endophytic plant relationships.
So plants are getting all these microbes out of the soil. The first source is always out of the soil. So you got a good soil, given time, the plants can get those microbes out of that soil. But how long is it going to take? I don't know. But the first source is the soil, and some of those microbes will be taken into the plants, into the roots, and then they'll move up through the plant to other parts of the plant. So, yeah, the soil can be extremely important tenet of regenerative agriculture right there. Build your soil up.
Yeah. Great.
Thank you so much, Walter and James. And yeah, thanks to everyone for your great questions. If you want to hear more interesting pieces of cutting edge research, I will say that the next session coming up at the top of the hour will be our poster session. So I look forward to seeing you there or somewhere else in our conference this week.